Hello and welcome to my presentation, Pathogen Inactivation of Platelets and Plasma to Improve Patient Safety and Well-being. My name is Marcus Becamero. I'm Scientific Affairs Director at Syros Europe. Generally, blood products are considered to be safe, and they are as safe as possible, but there is still room for improvement. Here we can see from left to right the transfusion-associated risks where pathogen inactivation can be a benefit. First, residual leukocytes. There is, even after leukofiltration, up to 1 million leukocytes left in a platelet concentrate. They could secrete chemokines and cytokines, which cause transfusion reactions, but also directly attack the host, leading to mild to severe transfusion reactions up to graft versus host disease. There could be new and emerging pathogens, like SARS-CoV-2, which we all know. It's not transfusion transmitted, but what if the next one is transfusion transmitted? Like we had with the HIV pandemic, which is still ongoing, by the way. The most common contamination of platelets is bacteria. The actual rate is 1 to 1,500, and this is more or less worldwide, as shown by multiple studies and publications. There are also screening limitations. If we have testing, mostly for hepatitis B, hepatitis C, and HIV, there is a window period. There are mutations, like occult hepatitis B, and there might be low viral load. And finally, there are known pathogens, like human herpes virus 6 or 7 or 8, which are normally not causing any symptoms in healthy individuals, but in immunocompromised individuals, they could be really problematic. And the majority of platelet recipients is immunocompromised. What we have to offer is the Intercept blood system, a technology to inactivate pathogens in platelets and plasma. Here is a brief overview how it works. On the left, we see a donor. He is donating, for example, apheresis platelets. They are contaminated by bacteria and viruses. We add our photoactive substance, amotosalen, which is a psoralen, which is not part of a natural immune system of plants. And this molecule is quickly penetrating through membranes and cell walls inside the pathogens and accumulating into nucleic acids. There, it's doing nothing until it's illuminated by light in the UVA wavelength. This is long wavelength UV light, which is not in the absorption spectrum of proteins, not causing collateral damage, but leading to a photochemical reaction, which cross-links nucleic acids irreversibly. There is no transcription, there is no replication. The bacteria die from apoptosis like cell death or necrosis. The white blood cells die from necrosis or apoptosis, and the viruses are replication incompetent. So now the platelets are safe for transfusion. The Intercept blood system consists of an illuminator, which illuminates the platelet units with UVA light for the photochemical reaction, and processing sets for platelets and plasma, which you see on the right. Briefly, there is one single sterile connection connecting the platelets or plasma to the processing set, which is a closed system. The component is floating through the red bag, which contains amotosalen, inside the first larger plastic bag, which is the illumination bag. Then we put it into the illuminator. We have a 5-6 minute step of illumination, and afterwards it's transferred into the CAD bag for platelets or through the CAD filter for plasma. CAD means count absorption device. It's a device to remove residual amotosalen and photoproducts. And finally, for platelets, it goes through a mesh filter into the storage bag. And for plasma, it directly goes into a storage bag. Here, we have a more detailed view on the platelet processing. First, the platelet product is sterilely connected to the Intercept processing set. Then, the platelets are floating through the amotosalen container into the illumination container. The illumination container, together with the whole kit, will be transferred to the Intercept illuminator. 2 units could be illuminated in the same time. Afterwards, the platelets are transferred to the CAT container. The CAT step takes, depending on the storage solution, 4 to, let's say, 20 hours. And finally, the platelets are transferred into the storage bag. The Intercept blood system is not a new technology. It's in use for more than 20 years. More than 10 million Intercept-treated doses have been manufactured globally. Intercept is the market leader for platelet pathogen inactivation with a market share of 87%. Intercept is standard of care in 11 countries. That means more than 50% of the platelets in those countries, often 100%, are produced with Intercept. The 11th country, which came that year, is United States. You can see the countries where Intercept is standard of care marked with the white stars. The first question when the system was developed, of course, is the safety for the recipient, the preclinical safety. Multiple safety and toxicity studies were conducted, which are summarized in these 3 review papers, which are, of course, publicly available. In the concentrations Intercept is used, it was reported that even 100-fold overdosing did not cause any toxic side effect. We see here a LD50 calculation for amoxicillin and paracetamol. In the first column of the table, we see the LD50 in red. It means the lethal dose where 50% of the rats will be killed. It's 1,000 milligram per kilogram of body weight for amoxicillin and 2,400 for paracetamol intravenous. Then, in the next column, we see the dose which is applied per transfusion for a patient. And in the third column, how we calculate this per patient kilogram of a 75 kilogram example patient. And on the last column, we come to the so-called safety margin. That means how much more I need to transfuse to kill 50% of the patients if they would be rats. Of course, for humans, there's no data available. I need to overdose 370,000-fold the amoxicillin, but only 168-fold paracetamol. So it's much easier to become intoxicated with paracetamol, which is, by the way, easily available. Now let's take a look at the bacterial safety. The average contamination rate, as reported from multiple studies around the world, is about 1 to 1,500. That doesn't mean that every 1,500 patient will develop a sepsis. The sepsis rate is more about 1 to 10,000 because not every bacterial contamination is causing a sepsis. The risk for septic transfusion reaction depends on the bacterial species, a lot on the bacterial load, but also on the immune status of the recipient. In an average treatment cycle, 6 platelet doses are transfused, which brings us to an average risk of 1 to 250 to receive a contaminated platelet unit during treatment, which is relatively high. We see here nicely the impact of different measures to prevent, or let's say reduce, the level of transfusion-associated sepsis. On the left, we have 3 examples of countries which, in the time point analyzed, did not do anything. No culture screening, no pathogen activation. We had high rates of bacterial transfusion-associated sepsis in Switzerland, France, and UK. The culture screening, we have 2 examples. In UK, it's much lower, 5.4. Compared to Belgium, 35, because in Belgium primary culture was applied, and in UK delayed large volume culture, which is more efficient, but the platelets are released on day 3, so you can only have them for 2 days. In countries where we had 100% pathogen activation with Intercept, in Belgium, Switzerland, and France, not a single case of septic transfusion reaction was reported. Recently, the British National Health System conducted a study to analyze the impact of platelet pathogen inactivation on product safety until day 7. They were asking the manufacturers to provide machines and processing sets. And what they did is they spiked, like many studies before, platelet units with different bacterial loads, inactivated them, but then kept them until day 7 of storage, because both manufacturers have a 7-day claim. And at day 7, they were analyzing the content of the whole bag, because bacteria could be unequally distributed for bacterial growth. What you see on the bars are the concentrations which are safe for 7 days. With Intercept, we see, for example, E. coli 6 log. That means 1 million bacterial particles per milliliter could be safely inactivated for 7 days. For the most important ones, according to the ECDC, Staphylococcus aureus, for example, or Streptococcus, we have also 6 to 7 log inactivation. Only Bacillus cereus was not inactivated by Intercept. Bacillus cereus is spore building and was likely building spores. No pathogen inactivation technology can inactivate spores currently. On the right, we see data for the only competitive platelet inactivation technology, Mirasol. In most cases, even with concentrations of 10 bacteria per milliliter, it was not safely inactivated until day 7. The authors reported that directly after inactivation, the cultures were sterile, but after the end of storage, bacteria were growing up again. That is also confirmed by data from national hemovigilance systems. Here we see data from France, Switzerland, and Belgium, countries having good and reliable hemovigilance systems. All 3 countries now treat 100% of their platelets with Intercept. In the past, that was different. They had only partial Intercept treatment or no Intercept treatment. And the data here is from 2005 to 2019. During this time, 3.5 million conventional platelet units have been transfused and 1.5 million Intercept. In the conventional units, 78 cases of transfusion-associated sepsis were reported, with 12 deaths. No case was reported with Intercept. That nicely points towards the prevention of septic transfusion reactions using Intercept for platelets. Now let's go to viral safety. The first question when it comes to viral safety is, which inactivation efficacy is needed? Normally you can say you need to have an inactivation efficacy, inactivating the whole viral load in the whole blood component or in the blood component, deducting the minimum infectious dose. Since the minimum infectious dose is often not known because it's really isolate specific, the efficacy is usually the viral load in the component. It's published that Zika virus could have 3 to 6 log, HIV 3 to 7, dengue around 5.5, but it's really different from person to person and from isolate to isolate. There is no guideline for efficient inactivation efficacy during blood transfusion. But there is a guideline from WHO and European Medicines Agency for the safety of plasma for fractionation. They consider a minimum activation of 4 log for a robust and reliable viral inactivation. Lacking other guidelines, that guideline is usually used by many blood centers to define a minimum quality requirement. However, it's possible to have deviations if you can show that lower inactivations are sufficient or high inactivation levels are needed. This is just in case there is not a lot known about the viral load and the minimum infectious dose. One problem is the transmission of tested pathogens. HIV, hepatitis B, and hepatitis C could be all transmitted despite nucleic acid testing and serology. There are multiple reports about occult hepatitis B transmission or window period transmissions. The Intercept blood system is the only technology showing high inactivation efficacy for hepatitis B, hepatitis C, and CMV and treponema in platelets. And except CMV, which is not needed because it's transmitted in white blood cells in plasma. Of course, it also has a high inactivation efficacy for cell-free and cell-bound HIV. Another challenge in blood transfusion are arboviruses. These are viruses transmitted by mosquitoes or ticks. The vast majority of infected individuals is asymptomatic. That means they are healthy, they may donate. Even they could have a very high viral load in the blood. The ferrets are not always effective because donors may give incorrect answers and the blood centers may not ask the right questions. It could also be that these viruses and mosquitoes are already endemic in the country they live. So even these people are healthy and they do not have any symptomatic disease, the recipient of such blood products could be in problematic health conditions with an impaired immune system, and the consequences could be disseminated severe infections, which are usually not diagnosed because people are very sick. Now they are more sick. Also here, the Intercept blood system provides a high level of safety against dengue, chikungunya, Zika, and yellow fever, the only technology for those viruses in platelet and plasma, and also for West Nile virus. Of course, using human coronaviruses is in the current pandemic always a topic. In the beginning last year, it wasn't clear if it could be blood transmitted, so there were high concerns, and of course we started some studies. Now we have a quite good data set on human coronaviruses. We have data for SARS-CoV-1 with 2 different isolates, SARS-CoV-2 with 2 different isolates, in platelets in 100% plasma, platelets in additive solution, and in plasma, as well as MERS coronavirus. Altogether, the data set indicates a strong protection of the Intercept blood system against human coronaviruses. But none of these coronaviruses has been shown to be blood transmitted until now. If there would be so many pathogen transmissions, why is there nothing known in my area? Usually there is a significant underreporting of transfusion-transmitted infections. They are often not recognized. The confirmation and validation is often too complex. They often occur too late. Later than 24 hours post-transfusion is often not considered transfusion-transmitted. And they are not reported because physicians are busy with other tasks and have a high workload. Now let's take a look at the inactivation of T cells. That slide is quite old, but still impressive. It shows the monitoring of the release of T cell-specific chemokines in platelet units. Pooled PRPs, which were not leukodepleted, were monitored for the release of interleukin-8 and interleukin-1 beta until end of storage. In black, you see the controls. They are untreated. And during the course of storage, of course, the release of chemokines increases in the back. After gamma radiation, it's less, but it's still happening. After Intercept treatment, there is no release. All T cells are safely inactivated. Until now, there are 3 published reports about the impact of the implementation of Intercept on the incidence of transfusion reactions. There is a paper from Switzerland reporting a reduction of 66% of severe transfusion reactions and 23% general transfusion reactions after implementation of Intercept. In France, there is a report of a 59% reduction of hypersensitive platelet-related transfusion reactions, the ones occurring in the first 4 hours after transfusion. And there is a study from Strasbourg in France reporting a reduction of 48% of platelet-related transfusion reactions and 39% of general transfusion reactions. Of note, a German blood center started last year or early that year using Intercept, and after a half year, they already recognized and reported a significant reduction of transfusion reactions. Of course, it's nice if we now kill all the pathogens and the white blood cells, have less infections and less transfusion reactions. But it's still needed that the platelets are efficacious, that they are doing their job, that they are not impacted by this treatment. The most accepted way to assess the impact of pathogen inactivation on platelets are so-called bleeding trials, which are also a requirement of the US FDA. Here, patients are enrolled in 2 arms, the pathogen inactivation arm and the control arm, and the incidence of grade 2 bleeding events is compared. For a statistical significant outcome, a larger number of subjects is needed, about 600, 700. So there are only 3 trials at all published which have a statistically significant outcome. 2 with Intercept, 1 with Mirasol. All these trials are analyzed in 2 analysis groups, like you should do it with the clinical trials. The intention to treat group, which contains all the subjects which were enrolled to the trial. It doesn't matter if they did get the right or wrong transfusions, if they participated all time or not, just enrolled is enough. This is necessary to prevent data cherry picking from the principal investigator. But of course, if many of these patients are off-protocol patients which did not participate correctly, it could be a problem. For this, we have another group, the protocol group. This analysis group contains only the patients which participated correctly in the trial from beginning to end, getting the right transfusions. For a good outcome, the results of both groups should match. We see here the results for the 2 Intercept trials. 0 means there is no difference between the arms in the incidence of grade 2 bleedings. However, we see in the Mirasol trial, in the per protocol group, there is significantly increased bleeding in the Mirasol arm. Not an intention to treat group, but a large number of patients were off-protocol patients getting the wrong transfusions. Another way to assess the efficacy of pathogen-reduced platelets are component use studies. That means the time span before and after introduction of pathogen activation is compared to assess the platelet use per patient of a specific indication. If I need more platelets for a specific indication afterwards, the platelets have less quality or less platelets are functional in the back. We have no increased use in 4 publications assessing this for Intercept, but in 3 for Mirasol there is an increased use. Especially the 2 Spanish ones have significantly more than 50% increased use. Pediatric safety is often a topic as well. Intercept platelets can be used for all age groups. Also for neonates, for infants, for pediatrics. That study from the United States was also assessing the incidence of transfusion reactions, means the safety, and there was no statistical significant difference between pathogen-reduced and conventional platelets for that group. In this study, also 10 neonates under phototherapy were transfused with Intercept platelets, and pediatricians were monitoring any kind of unexpected event or transfusion reactions, and no special event was noticed. Now let's take a look about the efficacy and safety of Intercept plasma. The thrombin generation assay is the most advanced technology to assess the quality of plasma, since it monitors the complete coagulation process from beginning to end. In that study from France, the thrombin generation was analyzed pre-treatment and 14 days, 6 months and 12 months storage after treatment with Intercept. Even after 12 months of storage, frozen storage, the values in the thrombin generation assay generated with Intercept plasma were within the normal range. Another topic which came up during the COVID-19 pandemic was the quality and quantity of antibodies after pathogen activation treatment. With the first attempts to make convalescent plasma, also pathogen inactivation of the plasma was considered to increase the safety because such donors are usually not normal donors and have a higher risk of intermediate transmissions. That study was the first one which was published for that subject by the German Red Cross. And what they assessed was first on the left, the quantity of antibodies against the SARS-CoV-2 spike protein with an ELISA. So on the left, we have non-pathogen-reduced controls, which were either stored in the fridge or in the freezer. On the right, after pathogen reduction, which were stored in the fridge or freezer. There was no statistically significant difference in antibody quantity. On the right, we see a plaque reduction assay to assess the quality. So here, different dilutions of the convalescent plasma were mixed with SARS-CoV-2 particles, and this was applied to tissue cultures to see if the particles are neutralized or not. Also here, there was no significant difference between, on the left, the control, on the right, the intercept arm, with respect to neutralizing activity. Multiple studies showed that the clinical safety profile of intercept plasma is comparable to conventional or karyotype plasma. There is also data for several clinical indications, especially coagulopathy in liver disease, liver transplant support, and multiple publications about therapeutic plasma exchange. Therapeutic plasma exchange needs multiple liters of plasma, increasing significantly the risk for infection because there are many different donors. Especially here, pathogen activation could be a big benefit. Summarizing the benefits for patients using amotosalen UVA-treated platelets units. First, we have improved treatment procedures. There is no gamma radiation of platelets anymore. All platelets are like irradiated. There is no mixing up of irradiated versus non-irradiated platelets, and there is a much lower incidence of transfusion reactions by inactivation of white blood cells in all units. We have a 7-day storage of platelets with intercept. That's especially for cross-match donors a benefit, which do not need to come so often. And there is less wastage, of course. And all platelets are CMV-free. There's also improved patient safety and well-being. We have significantly less transfusion reactions, 23 to 66%, depending on what is how reported. No transfusion-associated graft versus host disease until now, prevention of bacterial sepsis, and less complications and infections. Now we are facing a somehow unexpected pandemic, but this is not something which was not there the last 100 years. 100 years ago, we had the Spanish flu as a huge pandemic, causing millions of lives. But between that time and the current SARS-CoV-2 pandemic, there was the Asian flu and the Hong Kong flu, costing hundreds of thousands of lives. The HIV pandemic, which is still ongoing, which finally led to the development of diagnostics and blood transfusion. SARS-CoV, which was in Asia, bird flu and swine flu, the MERS coronavirus from camels, still endemic in Saudi Arabia, and Zika virus, which is now transmitted in Southern Europe locally. All these viruses were transmitted from animals to humans, and there's large potential for more. So we do not know what will be next, when it will come, how and how fast it will be transmitted, and how fast disease will progress. What we recognize is that with the beginning of a pandemic, the borders are closing quickly. There are no ways for engineers or application specialists to travel, and it's difficult to send goods because there is no freight, there are no planes. So it might be too late to implement a measure like pathogen activation to protect the blood supply when the pandemic outbreak happens. Thank you very much for your time and interest, and I'm now happy to take your questions.